everyone. So today we will be taking a look at research methods, some topics. So research methods understanding is crucial to scoring well in both paper 1 but especially paper 2. So let's take a look. So first we will be checking out samples and sampling technique. So sample is basically the group of participants that you're using in your psychological research. So the people that you experiment or research on can be human or animal. So the sample is then studied in detail according to the requirements of the research. There are three methods with which psychologists acquire a sample for their research, and these methods are known as sampling techniques. So the first sampling technique, or the first way to acquire a sample for your research, is opportunity sampling, where you simply use people that are available at the time of the research, so whoever you can find at that time. So for example, you're conducting a study at the train station, a field experiment even. So whoever is boarding the train at that moment, you can select some of those people as your participants, and that will be an opportunity sampling technique selection. The benefits of this technique is that there can be very little selection bias because the experimenter is unlikely to have much of a choice. He will have to make do with the people that he can find at the time of the experiment, and he cannot discriminate on basis of gender, appearance, or age. The second benefit is that it's easy to acquire because you do not have to look for a sample. Third is that it's more applicable because it's likely that opportunity sampling will be from your targeted population. However, it might be less representative because you can imagine if people are available at the same place and or at the same time, then they might be very similar. And these similarities that they share, it can make your sample overall less generalizable. So whatever the findings of your research, they will be less generalizable to other groups of people. The second type of sampling technique is a volunteer or a self-selecting sample. In this sampling technique, we acquire participants for our research by seeking them out ourselves. So we put out some sort of announcement or advertisement ideally. So benefits of this is that we are able to request participants with certain specifications or specific requirements. Usually we need these types of participants in biological research, and secondly, the participants are usually more dedicated to the research simply because they chose to come to us. Third benefit is closely linked to that second point. Third benefit is that because they come to us, it saves us some effort on our part. Problems, however, with this technique is there are potential selection biases. Because we have such control over who comes into our study, we might consciously or subconsciously impart some biases on gender, race, or appearance. There might be demand characteristics because participants who volunteer may have an added dedication to please the experimenter that they have signed up to be a participant of. The third is that you can imagine that participants who choose to be part of a study, who volunteer, are likely to share some similar traits or characteristics. These similarities reduce the generalizability of your sample and your study, and because it's less representative. The third and final sampling technique is random sampling. As the name suggests, in this sampling technique, we choose participants randomly. So everyone from your target population has an equal chance of being selected. So it's usually done when you have a list of names of as many people as you can gather from your targeted population, and then you randomly generate through some software or manual action. You randomly generate a list of names, and those names from this list are supposed to be your sample or your participants for research. The benefit of doing this is that it's likely to be more representative because you have no control over the specifications. So you cannot you cannot discriminate on the basis of gender, race, age, or appearance. Second is that it's possibly more generalizable because it's supposed to be more representative because again, everyone of every disposition or socioeconomic class, walk of life has an equal chance of being selected if they are part of your target population. A target population is basically the group of people or groups of people that you aim for your studies studies findings to generalize to. So if you're conducting a study for toddlers, or if you hope that your results are applicable to toddlers, then you would expect that your sample is also from toddlers. That would be your target population, and ideally, samples should be from your target population. There can be problems such as because I mentioned we can have no specifications in the qualities of our participants, so we cannot request specific participants. So usually this type of sampling technique is not employed in any biological approach research. And there might be less response rates because think about it. You generate a list of people from the names you have, but not all of them might agree to be part of your research. So unless you have informed them in prior, and even then they might withdraw from the research easily. So you might have to replace them, and that might be problematic. And third is that the representativeness or accuracy of your sample depends on the accuracy or representativeness of the list of names that you derive your sample from. So if the list has not been updated or if it isn't very representative to begin with, it is highly unlikely that the sample you draw from it, even if it is random, will be very generalizable or diverse. That So now here I have helpfully compiled the sampling techniques used in all 12 of our core studies in the AS psychology syllabus. So first we'll take a look at the learning approach. So in the learning approach, we have Bandura who used opportunity sampling where he had 72 kids aged 3 to 6. Then we have Saavedra and Silverman where we again used opportunity sampling where we happened to come upon a boy who's 9 years old and he had a button phobia. Then in Pepperbug, we also used opportunity sampling, where we simply selected a lab parrot, who was an African gay parrot called Alex. In the cognitive approach, we first had Andretti, who selected her participants through opportunity sampling, where we had 103 adults with an age range of 18 to 55. And then we had Baron Cohen, who employed opportunity sampling for his 225 normal adults and volunteer sampling for his 108 for his 1 and 5, so it's 15 autistic adults, male adults. Then we had Laney, who employed opportunity sampling for experiment 1 with 128 undergrad students and experiment 2 with 103 undergraduate students. Then in the biological approach, we first had Canley, who used volunteer sampling to request 10 healthy right-handed females. So this is what I meant by the specifications we can make when we use volunteer sampling. In Dement and Kleitman, we used opportunity sampling to select 9 participants, out of which 7 were male and 2 were female. In Schachter and Singer, we used opportunity sampling to select 185 male undergrad students. In the social approach, which is our final approach as well, we first had Milgram, who employed volunteer sampling to select 14 males for his experiment. Then we had Pilivin, who used opportunity sampling and because it was a field experiment, she simply used whoever was available on the trains on the time for experiment. So I believe there were around 4,500 participants. And then in Yamamoto, we again used opportunity sampling where we simply used any 5 chimpanzees that were available to us from the Kyoto University Institute. Then now we move on to another topic under research methods. This is types of data that can be collected. So essentially the types of data that can be collected during a research is broadly classified in these two categories. So first we have quantitative data, which is basically data in numerical form. So think of like scores or frequencies. For example, how many times a day do you brush your teeth? So that can be frequency, and what was the average score on a memory or eyes test? That would be all quantitative data. And you can collect quantitative data using any sort of closed-ended questions, but specifically rating scale questions. You can analyze quantitative data using central tendency measures and measures of spread. Qualitative data, on the other hand, is the complete opposite, so it is data collected in the form of words or just descriptions. So it's detailed accounts of information, and it helps us gain insight into the reasons of participants' behavior. And you can collect qualitative data virtually through any sort of open-ended questions in either open-ended questions or observations. So then there are certain benefits to collecting quantitative data, which it's more objective and it's easily comparable, which also improves validity and the reliability of your research. And this data can be represented graphically and it is easy to analyze. However, there might be limited response, as if you ask your participants to answer quantitatively, it is very likely that they will not be able to explain the full spectrum of their behavior or feelings. So this data often needs to be accompanied by more qualitative data to be understood better. Qualitative data, on the other hand, has certain benefits such as it's detailed, so it provides a lot of understanding and reasoning, and it's valid, and cannot be affected by outliers. So you cannot group a bunch of similar data in the same category and miss out one or the other, unlike you would in quantitative data. However, there are a few problems with this type of data because it's harder to analyze. There might be subjectivity when you're grading this type of data, and that subjectivity or bias can compromise the validity of your study. And it can't be too generalizable because the data collected is usually specific, especially if it is something like self-reports. So you can imagine how each participant's answers might be quite unique if they are given an open-ended. Now, there are certain ways of representing quantitative data. So again, numerical data, we can represent it through these graphical ways. The first is a pie chart and a bar chart. So this is what an average pie chart would look like, and this is a bar chart. Then we also have a histogram, which looks very similar to a bar chart, except there aren't these gaps in between the bars because they are all one category. So usually histograms are made on ordinal categories, whereas bar charts are made on nominal categories. So if you ever answer a question in paper 2, just keep in mind, according to the type of data you have, which graphical representation should you choose. Then we have a normal distribution curve. This is usually used in accordance with the mean, and ideally in the center of the normal distribution curve, you will have your mean, and on either side of the mean, you will have an equal amount of data. So it will be symmetrical. It's also known as the bell curve. And lastly, we have a line graph and scatter graph. So this is a line graph, and it basically shows you the relationship between two data points at a particular time. And it's helpful for noticing trends in certain things. And this is a scatter graph. A scatter graph or a scatter diagram is usually drawn in correspondence with a correlational study, where we have two measured variables and there needs to be quantitative data in both variables so we can see if there is a correlation or relationship. So it can be positive, like this is, or it can be negative, which would be a line like this. So this can, again, it's used in correlational studies, and this line that we will draw is called the line of best fit. So it is drawn along all these points, so wherever there are most points that fall on the line, we draw it most accurately we can. So these were the graphical ways of representing quantitative data, and in the next lecture we will move on to observation.